Funding for this program is provided by Annenberg CPD to advance excellent teaching.
The Human Genome Project delivered a breakthrough, the complete nucleotide sequence of the human genome, and the sequences of several model organisms. Now researchers are looking at the products of the genome on a large scale, using the tools of proteomics.
It's exciting because we now know enough about the basic cellular processes that what we're trying to understand are more complicated features, and proteomics allows you to get a handle on lots of different proteins at once.
How can proteomics help us understand the mechanism of drug design, or see disease before it begins? Proteomics holds the promise of important changes in science and medicine. Every plant, every animal, every microbe, every living thing we can see and can't see contains a collection of versatile and powerful proteins. Proteins serve important functions in all living organisms. They give the cell its structure, they catalyze its reactions. And when it's time for a cell to become a part of a blade of grass or the wing of a butterfly, it's the proteins that point the way. Now, researchers have a new way to study these crucial protein functions using the tools of proteomics.
So in studying any given process, biologists would always focus in on just those few proteins that we could assay and identify things going on with them. And now we can step back and we can investigate a process by saying what's happening to all of the tens of thousands of proteins that might be present in a cell. And that means you can ask very different questions than you could before.
Researchers define the proteome as the set of proteins in a given cell at a given time. Those proteins are constantly changing to adapt to changing environments, inside and outside the organism, making the proteome a dynamic entity. For many proteomic studies, research begins with investigating an individual protein's three-dimensional form.
The structure of a protein dictates what that protein is capable of doing. And if you really want to understand a process like DNA replication or like cell division, you really have to be able to look at those proteins at the level of structural detail. You have to be able to see which protein fits into another and which proteins bind to small molecules in the cell. And you can only do that through structure.
Most drugs work by blocking a protein's active site or changing its structure. If medical researchers can understand the complex three-dimensional structure of the protein, then they can design small molecules that precisely target those proteins, a process known as rational drug design. Because of this promise of new treatments and the potential for high revenues, it is the pharmaceutical industry that is fueling most of the research in the field of proteomics. But determining protein structure isn't easy. Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to coil and fold into unique three-dimensional formations. Two kinds of secondary structure units are the alpha helix and the beta-pleated sheet. The folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as the quaternary structure. Researchers would like to be able to understand the protein structure by looking at the gene that codes for it, but it isn't that simple.
If you know the primary sequence of a protein, at the level of understanding we have now, you cannot fold up that protein into a three-dimensional shape. And the reason is that there are billions and billions of permutations as to how that folding can occur. Computationally, it's an extremely difficult problem.
At Cirex, a drug discovery company in San Diego, California, researchers work to determine protein structures of potential drug targets. Ned David is the company's founder.
The goal when you're doing rational drug design is to use the shape of this protein combined with information about how a small molecule binds to it to design a drug to fit snugly, much like a key into a lock.
For DPP4, the primary...
The goal at Cirex today is to determine the structure for DPP4, an enzyme related to insulin production. To achieve their goal, Cirex researchers go through 4 steps: crystallizing the protein, determining its structure, locating a binding site, and designing small molecules to fit the site. Their first step is to grow the DPP4 protein in a crystallized form using a robotic system. The protein crystal consists of the same protein in its native structure, repeated many times in all 3 dimensions. By amplifying its form in this way, researchers have a better chance of revealing the molecular makeup of the protein. But growing protein crystals is a challenging process, in part because small changes in environment can cause proteins to unravel and lose their conformation.
Proteins are by nature not designed to crystallize. They have very disordered labile surfaces, and by altering the solution that the protein is in, we can often cause these unruly regions of the protein surface to order so that the proteins come together in a specific way to form the three-dimensional array that we call a crystal.
Once protein samples have been grown, a robotic system looks at each of the experiments to see which proteins have formed successful crystals. Good candidates are examined, then harvested by researchers.
We cut the little area out where the crystal is growing, and we then have to rescue very quickly that crystal from the drop before the drop dries out. We sort of scoop the crystal out, and then we freeze the crystal for shipping to Berkeley.
At Berkeley Laboratories' Advanced Light Source in Northern California, researchers use X-ray crystallography to help determine the structure of the protein. An X-ray beam is shot down a 100-foot tube passing through the crystallized protein. As the beam passes through the regularly spaced atoms of the protein, the crystal diffracts the X-rays into an orderly array, producing data that reveals the location of the atoms that scattered the beam.
So what X-ray crystallography has done is to provide us the structure now of many, many different proteins at a very precise level, at the level of angstroms, of tens of billionths of a meter resolution.
The data from the X-ray crystallography is transmitted back to the laboratory in San Diego, where computers construct a model showing the position of each atom in the DPP4 protein.
Looking at the xanthine ring now,
Researchers search for drug binding sites on the model in a process known as virtual ligand screening. Researchers first identify a site on the protein as a potential drug target. The protein model is exposed to a series of millions of small molecules. Each small molecule is chosen as a likely candidate based on its structure. When the computer finds a molecule of the right shape, it can predict that the molecule will bind to the appropriate site on the protein, either enhancing or disabling the protein's function. In the past, drugs were designed through a mostly random process that was costly and time-consuming. Without the ability to see binding sites, researchers experimented with many millions of random chemicals, hoping one of them would bind to the target molecule. But using a proteomic rational drug design approach, Cirex researchers believe they will be successful in creating a new treatment for diabetes.
We got the structure. We were first in the world to get the three-dimensional structure of DPP4. It is now our purpose in life to come up with the best small molecule drug that will bind with great potency and selectivity to DPP4 and switch off its biochemical function.
Three-dimensional protein models also allow researchers to see how a target molecule affects not just the protein it's binding to, but other proteins in the body. That way, scientists can see potential drug side effects long before the drug is tested in humans. In addition to offering advances in treatment, proteomics is also showing promise in diagnosing disease. At the National Institutes for Health, researchers are working on a test for ovarian cancer, a cancer that kills over 100,000 women each year. Elise Cohn is a physician and researcher with the National Cancer Institute.
Ovarian cancer is a devastating disease because we pick it up late. When patients are diagnosed in late stage, stage 3 or stage 4, there's about a 40% 5-year mortality, which means that 40% of women who are diagnosed with late-stage disease will not live more than 5 years, even with our best surgical and chemotherapeutic treatments.
And because the disease has few early symptoms, most cases are diagnosed late. Now researchers are using a new method that uses a proteomic profile to detect the cancer. NIH researcher Lance Leota is the study's principal investigator.
We imagined that as the blood is coursing through the tissues of the body, the blood ends up having an information content that reflects what's going on in those individual tissues, and that information comes in the form of patterns of proteins in the blood.
In previous days of trying to do this kind of study, you couldn't look at all the proteins at once. So what you could do is make an educated guess and say, perhaps this is the protein that varies in ovarian cancer, and you could design an assay, typically with an antibody to that particular protein, and say, does that protein change? But now you don't have to limit yourself to a single protein. You can say, let's look at all the proteins and find any protein that might be a marker.
Oh yeah, well,
so the big question was, which proteins in those thousands and thousands of proteins were the important ones that contained the pattern that was the signature for ovarian cancer?
In the study, blood is taken from patients with ovarian cancer and from women who don't have the disease. Serum is extracted from the blood samples, and small drops of the serum are spotted on a protein chip using a robotic system. The chip is then placed into a protein analysis device called a mass spectrometer. Inside the mass spectrometer, a laser hits the serum spots, ionizing the sample. The ions fly through the tube at different speeds, the ions with smaller mass traveling faster than those with larger mass. Using the order in which the ions hit the detector, the spectrometer determines the sample's composition and creates a profile of the individual peptide components of the sample. The end result is like a barcode, a display of the nearly 15,000 proteins in the serum sample. By comparing the samples from the women with cancer with the samples from the non-cancerous women, a pattern of proteins was discovered that appears to be unique for the ovarian cancer patients. Benjamin Hitt is the Chief Technology Officer with Coralogic Systems, a partner in the NIH study.
When we had all the data, what the computer algorithm was able to do was find a set of 5 features. When we looked at patterns based on those 5 features, we picked up 100% of all of the women with ovarian cancer, regardless of stage.
Right. Much additional testing will be required before this diagnostic tool is proven effective, but researchers and cancer advocates are optimistic.
If the promise of the screening tool is realized, then many, many more cases of ovarian cancer can be detected in early stage when it's treatable, and the outlook for women who have this disease will be dramatically, dramatically improved. I think ultimately it can save hundreds of thousands of women's lives.
Not all of the diagnostic techniques being tested will ultimately prove successful, but researchers speculate that diagnostics using proteomic profiles will become more and more prevalent. And eventually, this approach could lead to a different kind of visit to the doctor.
What a lot of scientists and particularly now biotechnology and pharmaceutical companies would like to be able to do is to take a profile of your proteins once a year and say, okay, we've just noticed that particular rise in the level of a certain protein indicates that you're in the early stages perhaps of a neurodegenerative disease, and we might be able to take steps early on to treat you for that before it becomes a serious problem. And proteomics is putting us in the position where we should be able to do that, not today, but at some time in the future.
At the Institute for Systems Biology in Seattle, Washington, researchers are also attempting to understand disease and the development of the human organism, but they use a different approach called systems biology. Leroy Hood was one of the key developers of DNA sequencing technology back in 1986. His work helped biologists break organisms down to their smallest parts. Now he's working to put them back together again.
Biology for the last 30 or 40 years is primarily focused on looking at one gene or one protein at a time. And although that's gained insights into biology, it hasn't really gained any insights into how very complicated systems work. Systems biology is an approach which must integrate many different disciplines, physics and chemistry and computer science and engineering and biology. And it's an approach that looks at the entire system. For the first time, we can take a biological system like the immune system, and we can interrogate, we can look at virtually all of its components and see how they behave with respect to one another.
A key element in systems biology is the creation of a predictive model, an attempt to formulate a description of how a system behaves.
The idea of predictive models has essentially two different components. One is that given any kind of input to the system, you can predict exactly how the system will behave. And number two, if we understand enough about the system, we can actually redesign it to create completely new kinds of properties.
The first step in creating a predictive model is to use all of the biology known in a system to formulate a preliminary model.
And then the second step is to do this systems biology analysis of that system, where we look at all its genes and all its proteins and all their interactions. But as we continually refine that picture in an iterative and integrative fashion, we'll go back and do more experiments and more analyses.
One of the systems ISB researchers are trying to understand is the sea urchin, a creature biologists regard as an ideal model organism for studying mechanisms difficult to study in humans.
Sea urchins use very simple developmental strategies, and hence they're enormously accessible. So what we can do is we can look at very, very simple systems that give us insights into the much, much more complicated human systems.
Hamid Balori is an ISB researcher who works on the sea urchin model.
And what we're interested in working at is how is it that you can start with a single undifferentiated egg and in this case over a period of just 72 hours end up with a fully functioning embryo that can feed and move and so on. How does that happen?
What we've done is identified the nature of the regulatory gene network that specifies how the gut of the sea urchin develops. And so now we have a network that includes 55 genes that describes precisely how across that 72-hour period the development of the gut of the sea urchin occurs. And in fact, we understand that sea urchin development so well that by rationally manipulating just one gene, we can create completely new types of systems. For example, by one manipulation, we created sea urchins that didn't have one stomach, but actually had two stomachs. And you could predict that accurately from the model.
Traditionally, people have thought of modeling as something that you do, it makes a prediction, and then you use that prediction, that's the end of the story. What we're doing is a little different so that the model grows with our understanding and kind of directs the next set of experiments that need to be done. It's a sort of a tandem process in which the modeling drives the experiments, then the experiments provide new evidence that we use in the modeling, and we just move on like this.
In this iterative process, gradually the model comes to describe the reality of the system with more and more specificity.
Hood believes that this integrated approach of systems biology will change research methods throughout the field. And perhaps in the years to come, Leroy Hood won't be just remembered as an inventor who helped take organisms apart, but as one of the researchers who helped put them back together. The strength of systems biology is that it explains the whole organism by describing the relationships that occur simultaneously within it. The regulation of genes, the production and interaction of proteins. Researchers studying yeast have created a functional map of protein interaction, describing the relationships between each protein. And even in this simple one-celled organism, the map is intricate and complex. But understanding protein interactions is crucial. It is at the core of understanding organisms.
Protein interactions are what underpin all of these complex machines that carry out the different functions of a cell. And the analogy that people often make is that of a three-dimensional jigsaw puzzle. That in the case of a jigsaw puzzle, you're trying to slot one piece into an adjacent piece to another piece. And the same thing is true in protein structures, that proteins must come together in highly complex arrangements where one protein fits into a complementary surface on another protein, then a third protein slots in, and a fourth protein slots in.
And over millions of years, protein interactions have been changing. As more and more complex organisms develop through evolution, the proteins have also evolved, developing more and different interactions. But that raises a question. As proteins develop new interactions, how do they maintain their crucial existing interactions? Protein interactions require an exact fit. But as proteins evolve, they might change and be able to form new interactions. However, even a tiny change in the protein structure could mean one of the crucial existing interactions is no longer functional, and that could put the entire organism at risk.
If you think of putting together a jigsaw puzzle, you're hoping that the accuracy of the manufacturer is to maybe a fraction of an inch or so, so that the pieces come together. But if you think of protein structures, they have to be accurate to the level of angstroms. That is a 10/1000th of a meter in order for those proteins to slot into each other. And if they aren't that accurate, then the complex can't assemble.
So how do proteins maintain their crucial functions in an organism even as they change? Some studies have theorized that proteins that directly interact with each other might be evolving at similar rates. But only able to study one protein at a time, no one had ever tested this hypothesis. At Stanford University in Northern California, Aaron Hirsch decided to use new proteomic and genomic data available to test this hypothesis. Hirsch and colleague Hunter Frazier looked at the evolution of individual proteins by comparing the proteomes of two organisms that have been evolving independently for a long time. Saccharomyces cerevisiae, brewer's yeast, and Caenorhabditis elegans, a nematode.
We took the yeast genome, we took a distant relative, the worm, and we asked how different is this yeast protein from its closest relative in worm. And we used that difference to estimate how fast that protein has been changing along its evolutionary descent.
Researchers are able to conduct this kind of study because at some point, hundreds of millions of years ago, the yeast and the nematode shared the same ancestor. Even though they've changed over time, they still have many similar proteins with similar functions. By finding proteins that have changed, researchers can track when they changed and by how much. But even with access to new proteomic databases, the study was challenging. And after nearly 1 billion years of evolution, just finding the homologous proteins was difficult.
A big problem when doing comparative genomics, comparing one genome to another genome, is that if they've been evolving separately for a long time, then the proteins in them can look very different. So it's hard to find the corresponding protein from one organism to another organism.
It's a little bit like trying to detect the remnants of the Big Bang in the radiation that we receive from outer space. There's very little of it there because it's been a very, very long time.
By searching gene databases and finding the best protein matches between S. cerevisiae and C. elegans, Hirsch and his team were able to assemble 4,000 protein interactions for study by a computer algorithm. And after many months, they had results. Research data began to show that proteins that directly interacted with each other evolved at nearly identical rates, while other proteins evolved at different rates. The results confirmed what Hirsch and his colleagues predicted.
Not just what's the evolutionary...
The use of molecular data to study evolution is creating new opportunities to understand the mechanisms of evolution.
Take the evolutionary distance from...
Molecular evolution underlies the more complicated kind of evolution that you think of when you think of apes to man, because when you think about it, what's going on in evolution is just you're seeing the changes accumulate on the large scale when you look at the large scale, but those changes are coming from the DNA level. So by looking at the DNA level, we're really getting down to the most detailed level that we can get to, and we can perhaps someday apply those findings to larger scale evolution.
The hope now is that we're going to look at many complex interactions among molecules and start to understand the way those complex interactions affect change at the level of the entire organism.
Aaron Hirsch and Hunter Frazier were able to conduct their study thanks to the new tools of proteomics. Without these tools, earlier scientists did their studies one protein at a time. Max Perutz spent nearly 22 years determining the structure of hemoglobin, and John Kendrew revealed the structure of myoglobin after working for over 12 years. Today, researchers are able to solve protein structures in a matter of weeks, and scientists are able to study not just individual proteins, but thousands of proteins at once. These new tools of proteomics are leading to a greater understanding of how proteins help create a living organism. It is the continuing breakthroughs in this field that will offer new possibilities in every area of biology.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.